Okay, welcome to the Biotech Rapid Fire Screencast. Um, Biotech, remember that we have lots of different tools that we use in the biotechnology world. We use restriction enzymes. We have molecular glue in the form of DNA ligase. We use gel electrophoresis to separate DNA by size. We use polymerase chain reaction, or PCR, to make billions of or millions or billions of copies of a single piece of DNA sequence. And then we have DNA sequencing that allows us to determine the sequence of DNA. So restriction enzymes are little molecular enzymes that cut the DNA. So they cut the DNA at specific sequences called restriction enzyme recognition sites, and they often leave behind a single-stranded overhang called a sticky end. So here's an example. You have DNA, you cut it with a restriction enzyme, you are left with two sticky ends, which have complementary base pairing, allowing us to easily put pieces back together with DNA ligase. So if you look here at the 5' to 3', remember DNA, always anti-parallel, 5' to 3', 3' to 5'. Here, whatever the overhang is on, this is considered a 5' overhang. Here's a very common restriction enzyme called EcoR1, and remember, restriction enzymes always kind of cut in this like zigzag pattern. If you cut two different things with the same enzyme, we can then use those overhangs to put them together into recombinant DNA, recombining two different pieces of DNA. Those are held together by complementary base pairing and hydrogen bonding, and then solidified by DNA ligase, which bonds the backbone, the sugar phosphate backbone, back together. So when we're creating recombinant DNA, we have DNA, we cut it. We have another piece of DNA. We cut it with the same restriction enzyme. We can then put those together. They will bind by hydrogen bonding, and then with DNA ligase, we will seal those together. Very often in biotechnology, we are using bacterial plasmids. So bacterial plasmid is just a small circular piece of double-stranded DNA that is distinct from the cell's chromosomal DNA, which remember is also a double-stranded circle. There are lots of things that are on plasmids that make it very useful for researchers. Many of these are made in the lab. There are some naturally found plasmids as well. So in a plasmid, you have to have an origin of replication, right? That is where DNA is going to replicate during S phase of interphase. Restriction enzyme sites, usually kind of all over, so that we can cut that plasmid and add different things that we would like. You need to have a promoter for that will drive expression of a target gene. And remember, promoters are necessary in order to transcribe the gene that is downstream from it. And then you have to have an antibiotic resistance gene so that we can select or figure out which plasmids have what we're looking for. So here's a really quick example of this. We have DNA on a plasmid. We're going to mix that. So this is for bacterial transformation, which is what we did in lab. Right? So we have DNA, our target DNA that is on a plasmid, and we are going to put that in with just bacteria. We're going to heat shock that bacteria, and then we are going to hopefully have about maybe 1% of the bacteria pick up that plasmid, right? So it's not very many of the bacteria actually have the ability to pick that up. Only about 1%. Right? These bacteria that have picked up the plasmid are considered transformed. But how are we going to get that 1% of bacteria, and how are we going to select for that bacteria? We're going to grow them on on agar plates that have antibiotic. Only that correct plasmid that has the antibiotic resistance gene will grow, so you get colonies. You can pick up that colony, grow it back up. Now we have a lot of bacteria that have that plasmid, right? Excuse me. All right, so gene cloning. The goal of gene cloning is first to cut, so let's say we want a gene from human cheek cells. excuse me, we're going to somehow get that out of that cell, which we're going to talk about in a second. We're going to cut that out. We're going to make many, many, many copies of it. We are then going to put it into a plasmid through, restriction enzyme cutting of both the plasmid and the gene of interest, DNA ligase to put them together, and then bacterial transformation. And then that bacteria actually makes the clones for us. So recombinant DNA is really important for a lot of things. So once we have a bacteria that is grown in a lab that has our gene of interest, we can do a lot of things with it. We can basically study the gene, or we can study the protein that it makes. So we can do both things in lab. So in lab, what we do is we first need to create recombinant DNA, just as I said. So we're making a plasmid. We then transform that plasmid into bacteria, select for it on antibiotic plates, and then we grow up lots of plasmid that is lots of bacteria carrying plasmid, and we use them to make as little factories to make the protein. So for example, if we were studying insulin, we could get the gene from humans, we could transform it into bacteria, we could grow up that bacteria in lab, we could get the protein out of that bacteria and then study the insulin. So there's lots of transgenic animals that humans have made from recombinant DNA. These are all kind of examples of this. One of the things that we use all the time in research science is having animals express the green fluorescent protein. This allows us to study individual proteins in a live animal. So a couple other methods that you have to know about: DNA polymerase chain reaction, gel electrophoresis, DNA sequencing, and nucleic acid hybridization. So polymerase chain reaction, here are the three major steps. We have denaturation, which separates the two DNA strands. That's at almost boiling. We have annealing, where primers are added. Remember, the primers are dependent on what piece of DNA we want to make many, many, many copies of. Those are sequence specific. We then have extension, where DNA polymerase will add onto the 3' OH of these primers to make many, many, many copies, or to make one copy. Then we do this 30 times, and we make 1 billion copies. So I just want to mention, do I have that here? That one of the most important things about the PCR is that we have to use a heat-stable DNA polymerase. Again, those are usually found in very, very hot natural environments, such as hot springs and that kind of thing. And that is because we need a polymerase that is not going to denature at 72 degrees. So a heat-stable or heat-thermal one. Okay, so PCR is used all the time in forensic science. So you can look at DNA electrophoresis gels to try to figure out who committed a crime, for example. And again, DNA gel electrophoresis separates DNA by size. But in order to look at it as a gel, you have to make millions of copies first. So in gel electrophoresis, DNA samples are loaded into a well at one end of the gel. Electric current is applied. DNA is very negative, so it will move towards the positive electrode. Larger DNA is slower than smaller DNA. Small equals fast. Gel is then stained with something that we can then look at the DNA. So again, at the beginning, you see nothing. At the end, this DNA, these pieces of DNA, are moved by size. Smaller equals faster. So DNA gel electrophoresis allows you to look at PCR products, make sure that they are the right size by comparing them to a DNA ladder, which is just DNA of known sizes. And again, you should be able to interpret DNA gel electrophoresis gels and understand what the sizes are and that kind of thing. You should also be able to look, for example, what we did in lab with our PTC tasting, whether something is homozygous or heterozygous based on DNA gels. DNA sequencing depends on individual nucleotides, but these are special nucleotides that do not have that 3' OH, so they are considered synthesis stoppers. And if we have lots of those, we can determine how DNA is sequenced. These are called di-deoxynucleotides. They stop synthesis. And then if you have all of those things stopped at different places and they have fluorescent tags, you can then read the DNA. So the shortest is A, meaning that's the beginning. The second one is AT. Then we have ATA, etc. So you're always reading sequencing gels from the bottom up. Nucleic acid hybridization, all it is using the power of complementary base pairing, which again is driven by hydrogen bonding. So you can look at the target gene by putting a little fluorescent tag on them. You can look in vivo, so like in living cells. And you can also use microarrays, which compare the expression of genes in two samples. And again, when I mean expression, I mean what mRNAs are expressed in normal cells versus cancer cells. We didn't talk a lot about southern, northern, and western blots, but I will mention them here. They're just different DNA, they're different electrophoresis to look at either DNA, RNA, or proteins, right? So we can look at different things on gels depending on what we are looking for. So kind of DNA, RNA, and protein. Southern and northern blots use complementary base pairing. Western blots actually use antibodies that bind specific proteins. So for forensics, remember that that all of our cells have mitochondrial DNA, and that mitochondrial DNA is derived directly from your mother, right? So in the original cell, your zygote, those mitochondrial were maternal, right? Nuclear DNA is found from both parents. All of us have varying amounts of repetitive DNA, and we can use that in forensics. We can also use what's called DNA fingerprinting, where you take some DNA, you cut it with a series of restriction enzymes, and they give you a particular pattern. Everybody has different patterns when we're looking at DNA fingerprinting. Don't fear GMOs. There's also whole animal cloning, which we didn't talk a lot about, but this occurs through nuclear transplantation. So you have a donor cell and you have you have a donor cell from some sort of tissue. In the case of Dolly, it was actually mammary tissue. And then you take an egg cell from an ovary, remove the nucleus. We're going to then put that nucleus into this donor cell. And then we can form an embryo from that, implant it into a surrogate mother, and you get a baby born, which is Dolly. And that's it for biotech.